Thank you. And we will move now to Laura Furlong, and to Laura. Yes, we will travel to Spain, in fact. Laura Furlong is from the Hospital de Mar Medical Research Institute and Pompeu Fabra University in Barcelona. And she will present us the DisGenet knowledge platform. And we thank her for accepting to give this talk. And I leave her the floor. Thank you, Laura.
Thank you. Thank you for the introduction. Can you see my presentation properly?
Yes. Yes.
Okay. Okay. Perfect. So, well, what I will tell you today, in fact, the challenges that we have already seen in the plant community are quite similar to the challenges that we face in disease genomics. So, and with DisGenet and the knowledge platform, we started working on almost 10 years ago, or more than 10 years ago. In fact, what we are trying to do actually is to try to address these challenges, providing an open access platform to actually make it easier to access and use disease genomics data. So, to set a little bit the framework, we all know, or we are aware that how genomics is transforming the treatment of patients and also the development of new pharmaceutical treatments, but there are also challenges in this area, and some of them are shown here in this slide. One is the large volume of the information that is being generating thanks to new sequencing technologies. So currently we have more than 500,000 variants, genomic variants associated to human diseases. And also we have this data silos problem in this field. So there's a lot of information. Data is in principle accessible with different levels of access right, but the information is scattered in very different repositories. Often these repositories are disease specific, and it's very hard to make the proper connections. And this also relates to the interoperability problem, so that we were talking about a lot in this session. So the data is sometimes not properly standardized, so this makes really hard for computer systems to identify the data and understand their semantics, and therefore make the proper connections among these data sets. And one of the other challenges that I want to mention today is that so this is a really fast evolving domain. So there are a lot of new findings that are reported in publications, and there is no way to put this data and curate this data and populate databases that are curated by experts. So the result is a lot of this information is still locked in publications, and this is really a waste of resources and efforts. So with DisGenet, what we actually wanted to do is try to address these challenges. And so this is a public platform that holds a comprehensive database that integrates and standardized information on disease-associated genes and genomic variants, in particular SNPs. The focus of the database is not disease-specific, but we aim to cover the full spectrum of human diseases, and also phenotypes that are manifestation of diseases and also of adverse drug reactions. DisGenet is one of the Elixir recommended interoperability resources. And in this way, it can be accessed and actually linked to other resources. And we expect with this to support a variety of applications, both in the area of genomic medicine and also in drug research and development. So a little bit on how we develop the database. So the information on the genes and the variants that are associated to with human diseases are extracted from different resources, and you have some of them here. Each of them is really focused or in particular diseases. Orphanet is an example of a resource focused on rare diseases, or you are specialized on data from animal models. So each one has its own particularities. We extract the information from them, standardize and harmonize them in a common schema. And we also identified information from the scientific literature by means of our text mining tool. So we are able to extract associations of genes and variants with diseases and disease phenotypes. So the current version of the database harbors more than 1 million associations between more than 20,000 genes. This more or less is most of coding genes, but also non-coding genes, and 30,000 disease and traits, so phenotypes. And this information is also, is of course annotated with community-supported standards and ontologies, and is annotated with other information from other resources. And we also provide metrics or scores that aid the user to filter or prioritize the information, and especially also we keep track of the provenance. So text mining is a very important thing to us, because it enables to extract all this information that cannot be found in the expert curated database and is still in publication. So the tools that we currently use allows to extract the gene disease and the variant disease associations, standardize them so that can be searched, analyzed, and especially linked to other data sets. And actually, in the current release, more than 70% of the gene disease associations are actually extracted by text mining from the scientific literature. And this is also important if we consider that when genomic variants are identified from the sequencing of individuals, from patients, a large fraction of these variants have no information in expert curated database. So this is quite important challenge nowadays. So our text mining, what we actually do is to identify the entities, the genes, genomic variants, and diseases, and how they are associated. These associations are also standardized by means of the disjoint association type ontology that you can see here in this slide, a snapshot. This ontology is also part of the semantic science ontology. And we also handle some linguistic phenomena, such as the ambiguity in the naming of entities, and this is really a key problem in the biomedical domain, and really affect the performance of text mining systems. And also we try to identify negated statements, and this is an example of how we actually display this information. So these are sentences that are extracted as evidence of a particular variant disease association, and we can identify those that have some negation cues. And this information is also summarized in an index, that is the evidence index of the association, that provides a kind of metric of how many supportive and non-supportive evidence each association actually has. So here's very briefly a schema, or a simplified schema of the database, in which I want to just stress the different annotations from standard vocabularies and ontologies that we provide for the diseases or the genes and variants, and also these metrics that we have introduced to actually help the user to make a better use of the data. For instance, we have the DistroNet score that can be used to rank the associations based on the available evidence. And also, as I briefly mentioned, we try to make the user so the user can easily find where this association was originally described, so the source database or the article or the actual sentence that represents or provide the evidence. So in the latest release of the database, we also provided new data sets that is about association between diseases and can also be used for different applications. So very briefly, this genotype-phenotype information can be explored from different perspectives depending on the use case. So if you are more interested on identification of potential drug targets or selecting genes for developing sequencing panels, you will like to use the gene disease association dataset. Or if you are more interested in variant interpretation, the variant disease association might be more suitable. So how this data can be accessed? So we provide different tools. So there is the web interface for easy access and exploration of the data. For programmatic access, we have REST and Sparkle APIs. And also in the new release of the Cytoscape app, the support for Cytoscape automation. So you can use your R or Python script to actually run programmatically queries over the Cytoscape app and also in our package. We also provide the data through the Elixir beacon and different data sets, views of the data or the whole database can be downloaded for local installation. And because we support community developed standards, so different resources or link to DistroNet or have incorporated part of the data in their own analysis or databases. And here you can see an example of Reactome that from last year uses DistroNet to actually annotate the pathways that they provide. And these are some of the other resources that actually connect with DistroNet. And I will stop now to allow for questions. So this is, of course, the work of many people in our group. And for questions or queries regarding DistroNet, you can reach us at this email address or contact us through Twitter. And thank you for your attention.
Thank you very much, Laura, and you are perfectly in time. We have time for a few questions. Questions are not typed actually in the Q&A, maybe. Yes, some are coming. The one from Mohamedou. I wanted more information about the text mining tools and resources you use. Are they available as reusable components of pipeline?
Yeah, so the tool that we use is a tool that we actually developed. It's called Be Free, and it's also open access. If you are interested, you can write me and I can give you the URL.
Okay, thank you. Is there a question from the panelists who have microphone? Yes, there is a hand. Sorry. So, do you have the microphone, Alban?
Yeah, thank you very much for your presentation. So it's a very nice resource. So I was wondering if you have any information on which interface is most used by machines, because you have a very nice interoperable resource. Do you have more usage by machines or by humans? And if it's by machines, which media is the most used?
What do you mean by machines?
By robots, by algorithms, queries.
Okay, Not really. Well, we really don't check the access of robots. So we know that some of the tools are more used or others. So by programmatic access or users, but we actually don't have these statistics. Yeah.
Okay. Thank you.
Another question appeared: what is the benefit of using RDF as structure for the data integration? From Pierre.
So I think the benefit for using linked data is actually to be able to connect with other resources. So some of other resources like Nextprot or UniProt, or in the past Campbell, actually connect with each other through the RDF implementation. But yeah.
It's really interoperability.
Yeah.
And Pierre is also asking, are you using some reasoning inference to enrich the data integrated? I also had a similar question, because you're using ontologies to describe diseases or phenotypes, and these ontologies have, of course, a hierarchical structure. So is it possible to make inferences? Or?
Not actually within this GenNet. So maybe through the RDF, if you have your own ontology. So we don't have the ontologies themselves in this GenNet, but outside it, I think it should be possible. But we are not actually supporting that within this GenNet. Yeah.
Okay. And if we want to download it, how big is it?
No, it's not big. It's not a big database. No, it's because it's all process and aggregated data. It's nothing to do with genomic data and omics data. It's really small database. Yeah.
Okay, so thank you. So it opens avenues. Thank you very much, Laura.